Okay, so we are continuing with our data tutorials, this time on XCT data. This will be presented as a team effort. I'll start, and my colleagues Anil and Maruti will also get to talk about their favorite parts. So, I'll go through the data types that we provide as part of MONet, then tell you how we get to those, how we extract those parameters and data from the CT data, and walk you through our workflow. Then Anil will give you a detailed walkthrough of the process of MONet-specific process of data processing segmentation using the program Aviso. We mentioned some other data output formats, options, and another software used, and then provide you a few examples of how this data can be utilized. And part of that will be Maruti's presentation on flow and transport modeling using CT data. Just to remind you of how we scan the cores in CT, we actually collect 4 data sets as part of MONet on every core. 2 lower resolution scans cover the whole height of the column with about 165 micro resolution. And these images are collected so that they have a little overlap. They are stitchable into a full height image. Kind of give you a nice overview of the soil horizons, the general texture of the column. And then we collect 2 higher resolution data sets at about 3-inch window, which is the diameter of the core, and that doesn't happen across the whole height, but from the top and bottom halves. And these are not stitchable, but they are high resolution enough to get good core network data. So the full core network analysis we describe are done on those high resolution data sets. So we provide both image data and numerical data from the CT for MONet. The image data looks like that in the top, the full height of the column, and the higher resolution data sets are those where we provide these CT constructed images and also segment the pore scale structure. You can see here images of the total porosity, connected pores, and the pore network model. And those are all provided for MONet users. And for numerical data, we extract pore sizes for all each individual pores, total pore volume fraction, and also we investigate the pore network connectivity. From the pore network modeling, we can derive these flow parameters, permeability, flow rate, tortuosity. We'll tell you more about those in detail. And we also estimate the wet walk density from the CT data, image data. This is a screenshot of a typical spreadsheet containing numerical pore data. You can see that the individual pore volumes, there are tens of thousands of data points typically in these individual pore volumes, pore surface areas, pore diameters are there. And so one can get, for example, the diameters and plot a frequency distribution for pore size distribution. Also, you can see that the pore network module estimated flow and transport parameters are listed here in the 3 axis directions. And this kind of data will also be a part of tomorrow's tutorial. We created this workflow of our CT-based process, starting with image acquisition together with the image reconstruction to get the 3D data. And then there are steps like image filtering and before the segmentation happens, which starts with the thresholding. And then we kind of have 2 workflows, one for the general pore sizes, pore volume fraction, and one for the pore network, connected pore network analysis. And this will be discussed in detail by Anil. Before we get there, I say a word about data reconstruction, so that to get the radiographs into a 3D reconstructed data set, we have to run it through a reconstruction software. We use the Nikon scanner's own reconstruction software called CT Pro 3D, and here's a screenshot of that software. You're looking at here at a very important step for the center rotation calculation. For these rotational imaging techniques, one always have to determine the center rotation to get a reconstruction, and that's an example of a 2D slice of reconstructed data. And so now we're going to go through the data processing steps from the reading the data into a VISO and doing all the steps such as cropping, filtering, and segmentation. So I want to introduce Anil K. Batu, my colleague. He's a material scientist here in EMSL, a VISO expert, and he spends a countless number of hours on MONet data.
Hello, everyone. Thank you, Thomas. Thank you for introducing me and easing my job with your explanation. So today I'm here to share our detailed analysis workflow that we are going to use for all type of MONet course. We created one standard workflow, which all these steps that Thomas gone all through that one. We are using the same thing for all type of MONet course. After doing the reconstruction, we import the data into the VISO software, which is provided by the Thermo Fisher. And this particular software is used for visualization. At the same time, we can do the analysis as well in that one. So after importing the data, we do the image cropping so that we will get the more precise information from the soil, not from the outside environment, such as outside air, those details. And then we do the segmentation, which is one crucial and important step in our data analysis, because that one, using the segmentation, we select only the pores, not the other organic matter or other material which present in the soil core. And later we do the pore volume fraction analysis, pore network analysis, and then finally visualization for the users in 2D or 3D or some animation point of view. So this is the screenshot of the VISO software that we are using for the MONet course. And okay, guys, can see my cursor here. So this the left top section is the various process and analysis steps that we are using for this MONet course. And the bottom part shows you their individual properties, and the right side one, which will display the analyzed results. This is one of the MONet data. I'm showing here. So this is one single slice. Here we can clearly see the what type of different phases that present in the soil. This is because as Thomas said, we did high resolution scan. Because of that, we can clearly see this type of various phases, and we can identify the micro pores as well. Here the pores are black, and the white particles are high density material or some minerals present in the soil core. Here, this data is after cropping, as I said, because the cropping will give you the precise information from the soil and region of interest that we are mainly interested in to get the more information about the pores. Because like while packing the soil into the column, so there's a kind of false porosity which will influence the pore characteristics and other porosity values because of the gap between the column and the soil. So this is not actual porosity of the soil. So we are avoiding all those details and getting only the more precise information, such as porosity and other pore characteristics, such as like volume, area, diameter, and those details. This is the same data set that I'm showing in a 3D view, or we can call it as isometric view as well. Here we can clearly see this is the crop volume, and we can clearly see how these are the cracks and the pores and the organic and high density particles, and there are some root things as well that it's really hard to segment those things, that I will tell you later. The analysis, we divided this analysis into two sections. The very first section mainly concentrates on the porosity analysis, and the second one is dedicated to the pore network analysis. These images is example of the pore network model. So we do the using the thresholding, interactive thresholding, we set only the pores. Then we attach various analysis like volume fraction and other details. We attach that one, and we calculate all those derived quantitative information. And then finally, to the same segmented pores, we attach the axis connectivity, which give you only the connected pores out of all the pores. and we can subtract the unconnected or isolated pores. Then we do the pore connectivity, which is helpful for us to get the flow and transport properties. So this is the slides that I already shown 2 slides. So here we are selecting the pores, which are shown in the blue, by using the interactive thresholding module. And here one thing we need to observe that this selection is based on the intensity range that the user provides. This range is mainly based on the grayscale intensities. Here, while we are analyzing the soil structures especially, so the color map histogram is kind of continuous bell-shaped curve, which is nothing but tells you that the various phases are overlapping each other. So in this kind of particular cases, sometimes we do select the pores along with the root, sometimes we not. With this intensity range, the total porosity or pore volume fraction is approximately 20%. Here, our main intention is to get only the pore information, because porosity and pore network will play a vital role in guiding the nutrients, water, and other soil matrix and soil organic material throughout the soil. And this is visualizing only the pores after the segmentation. And by doing the analysis, like volume label analysis, we calculate the volume, area, and diameter, area fractions of each and individual pore. We can do that. And also we can extract the same data into the Excel sheet. And here we can do all our calculations. For example, in this slide, we are just showing the pore size distribution. But we can plot the volume size distribution, area size distribution, and also the area fraction distribution. We can, and also by adjusting the bin size, we can play with that, all these distributions. And also we'll get all the mean, minimum, max, median of volume, area, diameter, and everything as well. And coming to the pore network model, this is after, as I said, after doing the thresholding, we'll get the pores to the same pores. We attach the access connectivity module, and then we do the image subtraction, which is divide the connected and unconnected pores in the same user software. Then we do separate objects. Then we do the pore network model. This is the example of pores that are shown in slides, and these are the only connected pores, and these are the unconnected pores. And these bottom 2 images, those are like connectivity volume, and this one is the ball and stick model of pore network model. So this image is only the pore connections that present in the whole pores. It's kind of a little hard to see because the connection is almost like 19%, which is nothing but 95% of the total pores are connected within the pore volume. So it's kind of a little hard to see the connected pores unconnected. So we separated the unconnected just for the visualization. So here we can see the unconnected pores with the connected ones. And this is the pore network model. Then we attach the pore network model to get the flow and transport properties. With this fluid viscosity and input pressure, output pressure, we will get the flow and transport properties of permeability, flow rate, tortuosity. Because these calculating the flow property is really important, because with this one we will get an understanding of how the water or nutrients will flow into the soil. And along with the flow properties, we will also get the pore details, which is of individual pore, of volume, area, diameter, and their XYZ locations, which are really going to help the modelers to create the simulation things for this one. And here the balls are the pores, and these kind of sticks, they called as the throats or channel lengths. And also we'll get all these kind of channel information, how much the channel is like in area wise, in diameter, and how much is that's length. And also we will calculate how individual one pore is connected to how many individual pores. We'll get that all those information as well. And also further we will get the flow in that particular channel as well. We can calculate that thing as well. And from here onwards, Thomas will continue his talk. So as I mentioned, visualization will create some animations, those things as well. So this is one of the animation that we created in the user software.
Thank you, Arnav. Yeah, there's other data that we can provide from CT are these beautiful animations that one that created by Anil. Here, you just saw the, maybe I'll go back a little bit. The isolated pores in red, the connected pore space in blue, and then they can be visualized separately as well. And then the image is now changing to the pore network model where the pores are modeled with a simple sphere. Their diameter is proportional to the pore size, and the channels, pore throats are modeled by sticks. This is a simplified model, but very useful for flow and transport modeling. We can also export the 3D data as a mesh, as will be also discussed some more later. The mesh is often used for example, computational fluid dynamics simulations. Another format is the stereolithographic file format STL that can be put into a 3D printer. So if we save that format, then you can imagine printing out of a pore network, out of a polymer for a micromodel, for instance. And for numerical data, for MONet, we do not calculate at this point the pore throats, their distribution and pore coordination numbers, but those are also can be extracted from the pore network data. And so for those of you who are interested in trying or learning this image analysis, there are some other software tools other than Aviso. Aviso, Visual Graphics Studio or VG Studio, and Dragonfly are widely known and used in the community. Unfortunately, they are all expensive and require a license. Although you can get the demo version or a light version to test it and decide. But we have some free tools such as ImageJ, Fiji. Fiji does have a plugin called Trainable Background Segmentation, WBKA, which is it's a machine learning based algorithm can do segmentation. And ParaView is a visualization software for 3D data, also free. And for modeling, I list a few options: OpenPNN, Force by pFlowtran that you will hear about from Maruti soon, and OpenFOAM. I think I have some more information on these as a reference. And so now to some examples. One I showed before in the other presentation, basically the direct use of the exported 3D segmented pore space or pore data into a voxel mesh to be used in computational fluid dynamics simulations that can simulate the flow of multiple fluids actually, and can be very useful for understanding biogeochemical reactions, processes. And then I'd like to introduce Maruti Mudunuru, who will tell you about XCT to pFlowtran.
Thank you, Thomas. So here in the example 2, what we are trying to develop is a machine learning workflow to upscale XCT data along with other types of MONet data sets, for example, the SEM EDX and other types of FDSR data sets to a larger scale to model at a continuum scale called the code called pFlowtran. So this workflow contains 3 steps. We call it as a soil twin, a digital twin representation of how do we collect data sets from MONet and how we reduce the uncertainty in characterizing the soils. So the left side is a physical space. So you collect MONet samples at various locations. And what you do is the first step, you have the soil experimental data for verification and validation of this digital workflow. In the step 1, what you do is you have 3D, you image the soil, you have 3D XCT images from millimeter scale, as Thomas was mentioning, for various slices across the entire core. And you fuse that with the SEM EDX information, which gives information on the chemical composition of the carbon and associated other elemental compositions such as silica, aluminum, oxygen. So once you have that information, you fuse that information to get an entire 3D picture of how the chemicals are distributed within a soil core. So you get 3D pore information along with what does the pore contain? So whether it contains considerable amount of carbon or type of oxygen, like whether it contains oxygen or where the minerals are located. So once you have that 3D pore and chemical information data, the second step is about pore structure characterization and using that information to estimate the corresponding permeability. So permeability is very essential as we want to know how the carbon is degrading within a soil sample due to hydrological stressors or different types of environmental conditions. So once you obtain the 3D pore structure here, that's in the second. Okay, that's fine. So once you get in the step 2, you see the pore scale flow simulation box, wherein which you use either an open foam, which does the computational fluid dynamics, which is an open source software on this 3D pore structure to get the fluid flow velocity fields within the specific aspect of that sample. So from this fluid flow, what we do is that we do upscaling. So since open foam simulations are very computationally expensive, because let me give a understanding of the type of modeling we do. So the 3D XCT images, each image is of anywhere between 1000 by 1000 pixels, and you have 1000 slices. So that's like a 1 billion voxels. So that's like 1 billion mesh cells if you want to entirely resolve that XCT image. And SEM-EDX is much more at a very high resolution. That is anywhere between 1400 to 1400 to 1400. That's more than like 3 million pixels. And you want to fuse that. That's like more than 1 billion cells. And you want to model that 1 billion cells, the fluid flow in that. So that is extremely computationally expensive. So what you do is you chunk out different parts of the core and model those specific aspects of the core and construct the fluid flow simulations representative of that core. So, and once you have that representative aspects of the core, so you see this part, so you see a box of upscale model wherein which a specific aspect of the box represents a much bigger aspect of the fluid flow simulations. So what you do is like, hey, like with this pore structure, what is the associated permeability of the sample, and you upscale it and through the breakthrough curves. So what you do is on the sample, you estimate how much is the fluid flow properties and associated tracer transport properties and get the breakthrough curves at a macro scale level. And your workflow will be able to predict and understand how these breakthrough curves happen. So we have been developing this multi-step approach of this digital twin, and these workflows will be made available for the MONet users on EMSL Tahoma. So that's our major goal. So tomorrow I'll be showing a certain part of this step 1 workflow, given XCT image, how you can use an open source software or Python set of tools to do the segmentation of the XCT image. So let me go into the specific aspects of the things that we'll discuss tomorrow. So this is a very small subset of the entire big XCT image. So you have an XCT image, a slice is of 1000 by 1000 pixels. What I'm showing is just only 50 pixels. So a small certain segment of that entire big subset. So you can see the grayscale image. And when we do a first segmentation cut, that's a binary imaging. So you have you're clustering into 2 different parts, the denser part and the lighter part. So you can see how the pore connectivity, let's concentrate on the white one. You can see the pore connectivity is nicely done from the left hand side to the right hand side. So if you do an experiment on this small subvolume, you can see a nice flow connectivity between the left hand side to the right hand side. So what we do in a modeling approach is assign material properties, saying, hey, pore is like having a high porosity and the black ones are very low porosity. So we assign material properties, associated permeabilities with pore spaces with highly permeable fields and non-pore spaces with low permeability fields. You can do a multi-fidelity modeling. So that is like, you can do a first cut of low fidelity modeling on the segmented XCT image. And you can also do a high fidelity on the full resolution of the XCT image. So you can assign a spectrum of permeability values, heterogeneous permeability values on the image and do that type of modeling. So that is, we can do that using open source code such as pfloatran. So to give a context, pfloatran is an open source multiphysics code that allows you to discretize at a very high resolution. You can go up till 2 billion pixels and solve that on HPC systems like Tahoma, and it is very well scaled. And you can solve process models like flow, single phase and multi-phase, along with reactive transport. That is very useful if you want to understand carbon degradation. How does the system degrade under different hydrological conditions? You can do that to a reaction sandbox. You can also incorporate thermal effects, like how if a temperature is changing under different types of perturbations due to anthropogenic activities, you can incorporate that to the energy balance. So you can all couple them in a sequential or in a coupled way and solve on a mesh with a very high resolution, such as the one which I've shown below, a small segmented volume or the entire segmented volume, and do this on the HPC systems. So you can install it on the laptop. You can also do it on the HPC systems. If you want to resolve it at a very pixel level, you need HPC system to do this. So, so just giving a taste of what does it look like. It is very easy to learn pfloatran. It's a simple set of decks. So you have the top part is like a simulation deck wherein which you show, hey, I want to solve flow. I want to solve reactive transport. How do I want to couple? I have some chemical reactions in the system. So how do I enforce that? What type of concentrations are in each and every pixel? You can give all sorts of information for that in a simple cards, and you can debug that in a easy way to build a suite of models for analyzing the data, for analyzing flow, reactive transport, and thermal balance on the XCT set of images. So this deck is focusing on the carbon degradation modeling using pfloatran. You can also do the same for the flow, reactive transport, and energy balance. Okay, so once you construct a deck, we need a mesh. So the mesh is like here we have a nice structured data. So each pixel is like a voxel for us, or like a mesh cell for us. And it is very straightforward to discretize the mesh and give that to pfloatran. So the left-hand side shows the mesh resolution. You can resolve till the micro scale of the XCT image, where each pixel represents whether it is a pore or not. And on the segmented image, I have constructed a mesh, and you can see the 3D mesh for this pfloatran image. So we have like 50 by 50 cells by 50 depth the cells. So that's like 125,000 cells. You can easily run this on pfloatran on your laptop, and this is extremely fast. You can do steady state transient simulations, as well as getting the tracer breakthrough curves. So what I'll be showing is a very small part of this. How do we do the segmentation? How do we construct a mesh and visualize this mesh in ParaView tomorrow? So the next step, I will ask Tamas to discuss the core network model.
Thank you, Maruti. I think I just wanted to highlight another third example of using CT data. I mentioned this work using OpenPNM. Aviso can do this, but OpenPNM is free. So by taking the CT segmented porosity into this Python-based software, OpenPNM, you can reconstruct the pore network or show the pore network, visualize it, plot it by different color by pore size, and then extract probability distribution functions for pore sizes, pore diameter, or pore throat diameter. And this type of analysis is very useful for simulating diffusion lengths, for example. The diffusion will depend on the connectivity of the pore network. So here we are actually comparing two soils where we are seeing that the longer diffusion length due to the less connected pore network basically results in a worse diffusion or transport. And this is interesting or important if you want to understand if diffusion limited organic matter decomposition kinetics. So basically soil respiration. This is a comparison of a wild and the cultivated agricultural soil, and the soil respiration appears to be reduced in the cultivated in this case due to its reduced pore network connectivity. Yeah, this is an open PNM based analysis, and yeah, this is why I have a little bit more information on these tools. So this is for your reference. Links to the software tools. ImageJ Fiji will be featured tomorrow in the hands-on tutorial, review as well, and also Fortran and OpenFOAM that Maruti just talked about. So you will get a chance to try to use those tomorrow. And I think with that, we were pretty quick, but we're happy to answer some questions. I have some extra slides if there are questions, but a lot of these will be covered tomorrow in hands-on tutorial.
Back to the one slide or two slides before where you showed the graph. I didn't understand that. Could you explain it a little more slowly since we have time? What's going on there? No, the next one. No, yeah. Can you say that again? How we're supposed to interpret this?
All I can say about this is that the normalized carbon uptake is plotted versus the water saturation, so the soil moisture content, and this is work by our colleague Jianchu Zheng, and it's I don't really understand these curves.
Oh, okay.
Not well enough to describe for you, but I think this curve shows a shift in the normalized carbon uptake versus the saturation, and also it seems like less busy, and that's due to the reduced pore network connectivity. That's what I understand. And, if somebody is more knowledgeable, please chime in.
Yeah, so I know Jianchu works on pore scale carbon metabolism, and so what I think she's showing there is water content within pores influences carbon respiration in slightly different ways, whether it is a natural soil or an agricultural soil. Okay. I don't know beyond that, but I think that's the take home.
Yeah, I know we have now data for more soils, and that might be more clear, but I don't have that slide here. I just wanted to highlight the applicability of that sort of data for looking at biogeochemical processes. Any other questions? Yes.
I have a question. At the start, you said that you took the pores and you do low resolution across the whole top,
Yeah.
or you do high resolution across a 3-inch subsection. But presumably they're quite heterogeneous. So if you take that 3-inch subsection and you shifted it up by an inch, do you get the same answer or do you not? And how does that like factor in? Why 3 inches for 38 microns and not 1 inch for 10 microns? Can you go that far?
Well, yeah, two questions there. One is so basically, in the CT where you have a square-shaped detector plate, we just by scanning a 3-inch width, so basically zooming in so that only the 3-inch soil volume is shown in the image, will maximize the kind of the soil volume that is being looked at and maximize the resolution. So we have basically a 3-inch by 3-inch cube that we have the soil analyzed, and if you would go above it, it would be different a little bit, or below it. But, we made a compromise of not doing 4 or 5 scans, doubling the data and the time to acquire it. We just said that we're gonna try to be, representative in a way that we looked at the top and bottom half, the same soil core, but there are, only I can confirm that there are differences between those volumes. Not always, but sometimes significant. So it's a sampling issue. But yeah, the size, the 3-inch relates to the fact that we zoom in on the core so that we include the full width. And for the 3-inch core, that's 3-inch, so you get this kind of a volume.
It's 38 microns, sort of the smallest size that you would ever want, or do you ever want to go smaller than that? Like, are there soil structures at sub-micron scale?
I would, yeah, I would like to capture higher resolution data on every soil. So that's why my SMT proposal used 1-inch cores. But there are drawbacks for that too, because the narrower the core, the more disturbance or deformation you introduce to the soil, and also you're sampling even less. But that's at that width you can capture 27 micron resolution cores down to that size. Marcus was first. Oh, okay. Yeah, go ahead.
So in your sample, you will have particulate organic matters, right? That you are not segmenting right now. So are you taking them with the pores or are you considering them as the solids?
That's a great question. It's sometimes we're not sure whether that that pore is filled with water, organics. It is, if it's really similar in the density, then we might include not just pore space in that segmentation. We know that from better data sets when we let's say we do a scan, micro CT scan on an aggregate 8 hours long scan, which is all the time we spend on these 4 scans, then we get much better signal to noise ratio, and we see water in the pores, and the organics stand out better. That's an issue, and that relates to the question that came up about segmenting the roots as well. That it's a lot of similarity, and so it depends on the system and how good data we are able to collect.
Actually, we found it with one of our treatments. So it was a drought treatment, and we did a CT scanning of that. And the porosity were more than where we have precipitation and stuff. So when we go back and we saw that since the particulate organic matter are not decomposed in drought, this is like shifting the pores in those.
Yeah.
So yeah, there's a lot of things that those particulate organic matter can change in terms of the pores that we are seeing. So if there is like a protocol in place to just exclude them, or
Well, if you can see them. So my experience is that image-based approaches to include and distinguish particle organic matter typically are micro CT because of their size and just the contrast, and then of course people consider staining them for better visibility in the X-rays. So honestly, I can't tell how much what fraction of particle organic matter we are capturing in the pores in this data. That's
one more question I have, like the Shiny app that you have already have, do you have like variables from your pore scale analysis into the system right now so that you can see their like relationship with the type of?
not right now, but I would love to have the pore scale data in there. Yeah.
Actually, there are the summary metrics in there. So there, yeah, there's pore connectivity, tortuosity, porosity, and some other summary metrics as well.
Sorry, my mind was going to the images. It would be so cool to see the organics in the image. Like the CT, and that's but yeah, not that. It's a bulk information versus the images, and it would be nice to see everything in its place. Their concentration, but yes.
I'm curious how this relates to the talk we had earlier, Alexandra's talk. Can we do micro CT XCT here, or is that you need a different?
EMSL have one CT. There's a nano CT, which is more like a soft X-ray microscope, so it's not really applicable to soils. PNN does have a few micro CT scanners, and we did use the one in 3410 for some soil samples, including user proposals like Marcus Perry's project, but it is limited to size. So even though it's a micro CT, it also means that the sample size is much smaller. So if you want to get 2 micron resolution, then your sample size is basically 2-3 millimeter wide. The same rule of thumb, 1,000 of the largest diameter applies, but some of those systems allow you to do a region of interest scan. So you have this kind of a scoping scan, and then it co-registers to a zoomed in area to collect high resolution data. So you actually have this co-registration between the data sets that has to be part of the software. That's very powerful, I think. So for MicroCT for MONet will be through our collaboration with APS, with the synchrotron.
Will they be able to look at the pores and check to make sure that there's organic matter there or not, or
Well,
how far it is from the?
yes. At least at the synchrotron, you can play with the X-ray energy. So maybe you can do either dual energy scan and subtract data, and or stay in the organic matter if we are going after those. But on any CT tool, it's a challenge to distinguish the organic matter from, for example, in a wet soil or a wet soil sample. But it's a good goal, challenge. Yes.
I'm curious how long it takes to measure each soil core here for MicroCT.
Well, so we opted for settings where these kind of low resolution scans are done in I think 1.5 hours each, and then those 2 scans we spend 2.5 hours on. So it's 8 hours of scanning per core right now, and so that it can be programmed as a batch. You put in the core, and then you run. Even you can stack 2 on top of each other if they are nicely packed. And but back to the previous question, if you were to obtain this data on the whole core, it would be 5 2.5 hour scans just for the high resolution, and then you have issues with the throughput. And of course, person time and the data to analyze. But so 8 hours is the short answer for these 4 scans.
I have a question for Maruti.
Yes.
So Maruti, have you been able to replicate those breakthrough curves that you showed on the right using your digital twin soil?
Oh, so that part of the workflow, so I still did not match with the I still did not see the exact data yet. So that's the ultimate goal. So that part of the step is using a synthetic data, like through a dash part. So I was able to do that, but that's 2 years down the lane.
Okay, yeah.
That's like the step 3 is the next year. The first and 2 fusing the XCT and SCM EDX and getting the high resolution permeability is at least for this year.
Thank you, everyone.